Hello. Hi, guys. We're going to kick off in the interest of time. Me, Kayla King, and Zen Lewis wanted to organize a symposium to bring together some of the people working on ecology and evolution, mixing the sort of single microbe-host interactions with the microbiota interactions. And our first speaker has come all the way from Emory. We have Nicole Gerardo, who's going to be talking about environmental acquisition of ecologically important microbes. So please take it away. Do we have a talk, guys? There we go.
There we go. Otherwise, we're just going to wing it. So thank you so much for inviting me. This has been a great opportunity to meet some new people and see some old friends. Today I'm going to talk about one of the systems that we work on our lab. And in general, I'm interested in host-microbe interactions and sort of their ecological and evolutionary contexts. We work both on hosts and pathogens and host and beneficial microbes. And in some ways, it is different flavors of the same thing because all of these associations have costs and many of them have benefits. So I want to mention a number of people in my lab that contributed to this work. In particular, Tariq Acevedo, who is a former undergrad and now a technician, and is an amazing scientist and colleague. And without him, most of what you see today would not have been possible. So like any organisms, pathogens are going to be selected to maximize their fitness. And the main barrier we think about for this is the host. The host is going to mount immune response. There's going to be strong selection on hosts to try to avoid or attenuate the impacts of pathogens. And as we all know, that the environmental context matters. So host nutrition matters, temperature can matter, how frequently the two organisms contact each other can matter. But increasingly, one sort of part of this environment that is seen as critical is the microbiome associated with that host. And we know a number of systems where microbes confer protection to their hosts. These are 4 examples of gut microbes providing protection. In bees and in mice, we can see protection against protozoan parasites. And in locusts and in humans, we can see protection against bacterial pathogens by microbes within the gut. Now, it's not just sort of bacteria, which is true of the cases that I showed you, but we know that fungi play a critical role in protecting hosts as well, and that can be true in plants. So this is one of my favorite examples. Betsy Arnold works on fungal endophytes within leaves of plants. This is cacao. And the fungal endophytes can confer protection against both pathogens and predators of many plants, including cacao, including this fungal pathogen that you see on the right. This has important consequences for the holiday season and for my life as a scientist in general. I need to protect my chocolate source. So microbial conferred protection has both indirect and direct benefits on your lifestyle. Now, when we think about pathogens, there's going to be sort of 2 microbe communities that are going to be critically at play within hosts. One is their actual host that the pathogen is attacking, and for many pathogens, it is going to be the microbiome within the host that vectors that pathogen. We see a number of examples where bacteria within insects alter the capacity of their vector to vector a pathogen. And in both of these cases, the tsetse fly and mosquitoes, these are intracellular bacteria that decrease, in most cases, the capacity of the insect to vector the pathogen. But there are some cases where actually intracellular symbionts increase the capacity of insects to vector pathogens. What we know is that these interactions are incredibly complex. Whether we're thinking about the host or the vector, that the microbes may have direct effects on the pathogen through competition for resources, through toxic-mediated interference, because they're microbes. Microbes live in a competitive world, and they're going to evolve in response to that competition. But these microbes are also going to have indirect effects, either through changing the host immune response, or potentially for a pathogen that could be a beneficial indirect effect. If they increased a vector's fitness, then the pathogen transmission dynamics could be altered in a way that benefits the pathogen. Adding complexity to this picture, when we use the term microbiome, it is a complex term that represents maybe the presence of a single species that matters, the composition of the community, abundance of particular members, overall diversity of that community, or particular genotypes within species, or genes just in the community in general. And for a lot of animal hosts, this is a very complex picture. So this is a drawing from the New York Times, sort of a phylogeny representing the bacterial diversity found within you and most animals. And we know that there's a complex community in our guts, on our skin, on our tongue, on our teeth, and that community differs between individuals, between sort of periods of your life, and can be influenced by a number of environmental factors. And this community is so big and so diverse that this may actually be an inaccurate picture. A better picture may be something like this. Well, if you're a community ecologist, this is incredibly exciting. If you're trying to do experiments, this is incredibly frustrating. And so many of us continue to try to work with sort of simpler invertebrate systems. And many of those invertebrates that are best known for their association with microbes and that have been experimentally tracked are insects. And part of this is that we stand on the shoulders of some amazing scientists who came before us. So this is a drawing from Paul Buchner's work. It shows the gut of a true bug species, and you see these very invaginated regions along the gut. And he hypothesized that these structures, or similar structures that he found in a lot of insects, were there to harbor beneficial bacteria. When he looked inside these structures, he could see sort of one or a few morphotypes of bacteria, sometimes at incredibly high density. When associated with the gut, he often hypothesized that these likely had some nutritional benefits. He also hypothesized in this beautiful book that a lot of these microbes might confer some protection towards disease. Now, he didn't have a large experimental toolbox. He had no molecular toolbox. But just as if you sit and read The Origin of Species today and find that many components indeed have been supported by experimental data, much of what Paul Buchner hypothesized has been supported by elegant experimental work. So that we know a lot of insects are hosts to beneficial microbes. And then we can start thinking about how this plays a role in a system in which an insect host vectors microbes. So today I'm going to focus on the squash bug, Anassa tristis, that is a vector of a bacterial pathogen. And we're going to talk first about sort of its association with a bacteria that's found in its gut. And then we'll talk a little bit about interactions between that bacteria and the pathogen that it vectors. So this is Anassa. This is a huge agricultural pest, particularly for organic farmers in North America and in Georgia, where I live. They hate it. The end of the squash season comes in Georgia because of this insect, because they do huge damage on plants, which is somewhat frustrating when you're working with them in the lab, but makes them sort of very experimentally tractable at the same time as we can get large population sizes. They vector a bacterial plant pathogen, strains of Serratia marcescens. The pathogen can overwinter inside of adult individuals. We're not actually quite sure where yet. And then it's transmitted into the next field season, causes the plants to wilt. If we look inside of an individual, whether male or female, here we see the gut, and here we see sort of this highly invaginated section of the midgut that's very similar to that drawing that I showed you by Buchner earlier. When we use high-throughput sequencing to characterize the microbial communities in different sections of the gut. Here in a turquoise color represents sort of the bacterial communities within this specialized invaginated region, which I will refer to as crypts. And you can see that those communities within those are distinct from communities that we find in other sections of the gut. When we look at what species are there, we see a number of different bacterial genera within the crypts, but it is dominated by Burkholderia. These crypts are dominated by Burkholderia in every adult that we have sampled, and this bacteria is cultivable. This is actually not surprising. Yoshi Kikuchi, who's a scientist in Japan who's done some elegant work, surveyed a number of true bug species across a number of families and found that Burkholderia associations are quite common for insects within this very large, diverse group. And so the colored families here are ones in which species are consistently associated with Burkholderia. And indeed, the Burkholderia strains that we isolate from squash bugs, which are indicated with black circles on this phylogeny, are very genetically similar by a neutral 16S gene to strains isolated from other species, which are shown in the white circles. And this clade has become known as the insect-associated Burkholderia clade. There are environmental isolates within this larger group, but it is definitely distinct from Burkholderia that many of you may have heard about, as Burkholderia is actually a common model because it is found frequently in the lungs of cystic fibrosis patients, and that would be at the clade near the top. I mentioned that the bacteria are cultivable, which means that sort of opens up our toolbox quite a lot when we want to study this interaction, not just from the perspective of the host, but also the microbe itself. So we can fluorescently label these bacteria, and what we see is we see this sort of concentration of these bacteria within these crypts. So the crypts here are green, not because of autofluorescence, but they are filled with bacteria. If we zoom in on the crypt, what we see is this really structurally complex community. So you can see patches within this where the Burkholderia are concentrated. You can see other patches where they're not present. Interestingly, some recent work being done of models, sort of in vitro models of lungs of humans and looking at sort of colonization and sort of spatial organization, you see the same pattern. So that these sort of places where bacteria colonize, it's not like there's a bacteria every place, but you get these complex communities, and I think that this is really exciting from a community ecology perspective. So where are they getting these bacteria? To do this, we can feed young individuals or nymphs on diet that has the labeled Burkholderia in it. We rear them to adults, and then we can put them in cages. So the only way that the offspring of these adults are going to get the bacteria is either if it's directly transmitted from adult to offspring, or if it escapes these adults into the environment, and then they subsequently pick it up. So we checked sort of for subsequent offspring produced in these cages and set up 4 different cages. And what we find is we never find the bacteria on eggs, regardless of whether we sterilize them or not. So they're not in the eggs, they're not on the surface of the eggs, and we also never find them in first instar individuals. What we find is we start to find the bacteria within the second instars and later offspring of these adults that are harboring the GFP-labeled bacteria. And so this would be very consistent with an environmental route of acquisition. Mom is not giving it to her offspring, which is a very common trait across many different diverse insects and animals. She's instead, it is getting released from adults into the environment, and they're picking it up from the environment. So far we see a tight association between Burkholderia and squash bugs. And now, to really look at the effects of these bacteria on the bugs, we can first think about sort of impacts on development and survival. So here we have fed individuals with a strain of Burkholderia called SQ4A. because you go in the field, you isolate a bunch of stuff, and then you're stuck with the name for the rest of your life. I say SQ4A all the time now. Or we feed them on water controls, and then we look at survival and development rate. So here in green are the bugs who have acquired the Burkholderia. Here in black are the bugs that are fed on the water control. They've now been put onto plants in sort of hydroponic cages to reduce the likelihood that they pick up some other Burkholderia strain. And what we see is significantly higher survival of bugs that have Burkholderia. And they're also surviving to sort of the different instars. And indeed, in this particular experiment, when they're on plants, we found no individuals making it to adulthood in the absence of Burkholderia. So this is a critical symbiont. What we have found in subsequent experiments, if we give them sort of a high nutrient source, so if we give them squash fruit as well as plants, which during some parts of the season, the bugs are going to be able to feed on squash fruit, we can get a very small proportion of individuals to adulthood, but they are small, their guts are poorly developed, and we would assume that they would actually have pretty low fitness. What we also see is that they develop much faster to adulthood when they have Burkholderia. And this sort of begins in sort of the early instars, and then the time delay gets larger and larger between each subsequent instar. So this is a system where the symbionts seem to have critical positive effects on survival and on development, and yet the mother is not transmitting it directly to her offspring. As a mother, I think this is a really stupid idea. We know that many insects have evolved, including insects sort of within this larger group, have evolved mechanisms to pass bacteria onto their offspring directly, either within the egg or on the egg. And so it leaves the question of sort of why this is happening, and I cannot answer that question of why they do this. But we can start to think about the consequences of environmental acquisition versus direct vertical transmission. And one of those consequences is that if you're picking it up from the environment, there's the opportunity to pick up other strains of Burkholderia. So we've done an experiment where we fed insects alternative strains. In this case, you're going to see data from SQ4A again, which we know is highly beneficial, and SQ6C. So again, we feed them, track development and survival. And what we see is that SQ6C provides no significant benefit over not getting Burkholderia at all. We again see sort of the benefit in terms of survival of getting SQ4A. And the SQ6C also provides no significant benefit in terms of increasing development rate. We do know that SQ6C actually establishes at very high numbers. So one possibility when I saw these data is that they feed on the bacteria, but the bacteria can't establish, and then it can't provide a benefit. But it actually establishes at a higher load than SQ4A, yet provides no benefit to the insect host. So we see many ways in which these symbionts can affect the insects, and that is going to be symbiont genotype dependent. Now, another way that symbionts alter sort of their host and can then have indirect influences on a pathogen that they vector is by altering the insect's response to pathogens themselves. So to look at whether Burkholderia could influence survival of insects when they're exposed to a pathogen, we took individuals, we either fed them Burkholderia or not, and then we injected them with pathogenic Serratia marcescens. So Serratia marcescens is the bacteria that they vector to plants. It is also a bacteria that is a common insect pathogen and has been used in a lot of sort of ecological immunology studies to assess pathogenesis and sort of how environment influences host-parasite interactions. What we see is that when individuals have Burkholderia, which is in the green, that they have a lower pathogen load. And this is true within an hour of injection, and then 18 hours of injection, the pathogen load has actually gotten quite large, and this does have deleterious effects on survival. So there's sort of a multiplicity of effects of this symbiote. We believe it, based on the survival and development data in the absence of the pathogen, that it is providing nutrients or resources to its host. And then in some way, either that indirectly affects association with this pathogen, or there's direct interference in some way between Burkholderia and the pathogen. So now we can start to think about sort of more direct interactions between Burkholderia and the pathogen that is vectored. Again, this is the pathogen, Serratia marcescens. And we'll do co-infections and single infections of Serratia and Burkholderia. So in this case, the bugs are feeding on Serratia or Burkholderia, or be given a dose of both bacteria at the same time. In this particular combination of strains, these bacteria get to about the same sort of load within the insects when doing single infections. And during the co-infection, Serratia is eliminated. If you're a farmer, this is very good news. There are ways in which at least particular Burkholderia strains could limit the vector capacity of these insects. But if we look at two other strains, these happen to be two strains that get to much higher titers during single infections. Serratia actually outcompetes Burkholderia. And I think that this is sort of a common picture that is arising that comes from host-parasite interactions, that there's genotype-genotype specificity, and that many in these microbe-microbe associations, where a microbe is protecting a host or changing vector capacity, there could be very strain-specific effects. So now, briefly, what are these effects? So if Burkholderia and Serratia co-infect, and the Burkholderia leads to clearance of Serratia, is that through some sort of host response to the Serratia, or is it through some more direct competitive or interference mechanism? So to do that, we can take some of these same strains and look at sort of in vitro competition. Take the host away and see what happens. So in this case, we will grow the bacteria in isolation or together, but in a test tube. And what we see in this case is that these two bacteria, again, which are ones that you saw before, get to very similar titers in the test tube, and Serratia is lost. And these were the strains in vivo where Serratia was also lost. So this would be consistent with the possibility that these microbes sort of undergo some direct competition with the host that is not host-mediated. So overall, when we think about sort of how these microbes are influencing the pathogens and the pathogen's vector, we see a complex picture, that Burkholderia increases the ability of the insect to survive and develop. That's good for the pathogen, but potentially the Burkholderia may also alter the ability or lessen the ability of the host to vector the pathogen. And with that, I have a number of people to thank, and most notably the National Science Foundation that funded this work. Thank you. In a lot of communities that order of precedence matters. Yeah, I realize I should stand near the microphone probably. The question is about when they are picking these up in nature. So in terms of the symbiote, the data that I showed you with these cages is very consistent when we look at field samples, that we don't find Burkholderia in first instars. We find it in some second instars. We can't really age them, that we just know they're in that instar. And then we always find it in the field, in the adults. In terms of the pathogen, this pathogen has actually been very little studied in terms of the dynamics of when it's being picked up. And it is an epidemic, so you see it in some fields or another. And so we have not been fortunate enough to be able to go screen into epidemics and see what instars that we find it in. But I think there is a possibility that the mechanism that sort of limits Burkholderia getting in the first instar may not influence when Serratia gets in. And that's because Kikuchi, I mentioned before, in another species, has shown that that is really about gut development and that crypt developing. So the hypothesis is that crypt is just not developed enough for bacteria to get in it. So Burkholderia may be coming in. I don't think they're feeding any differently. It just doesn't establish in the crypt and then sort of gets wiped out. And then in later instars it does. With Serratia, our preliminary data suggests that it's actually establishing more in other sections of the gut, and so it may be able to get in earlier. Yeah, so I think the interesting thing about is sort of how is selection acting on these microbes. So from the perspective of Burkholderia, I don't know that association with squash bugs is really influencing the evolution of these Burkholderia in the population as a whole. So I think probably once they get in there, there's enough generation times that we see sort of evolution, and then they get out, but there's a lot of Burkholderia in the soil. With Serratia as well, I am not convinced by the data that's out there that this is the only vector, right? So it may be, yeah, there's a consequence of harming this vector if you clear their symbiote, but a lot of pairings, you're not gonna do that. And then if there's another vector out there, that's also going to sort of relax selection to there being a huge cost to that. And that we really need to tease apart. Thank you.
Okay. Thank you very much. Our next speaker is Steve Patterson from the lovely city of Liverpool. And he's going to be talking about microbial ecology in the CF lung. Thanks, Steve.
Thanks to Ellie and the organisers for the opportunity, and it's great to be following such a stimulating talk. So in a slight departure, I guess, for a ecology conference, I'm going to be talking about some clinical data and thinking particularly about ways in which one can view infection as being ecological or evolutionary process, and how we can apply some of those ideas to a clinically important infection. So let me start by introducing the collaborators here. So this is particularly what was started by Craig Winstanley here at Liverpool. He's a microbiologist, and along with Mike Brockhurst now at Sheffield, and Ellie, David, and Ben, who are the postdocs doing all the hard work, plus the NHS and the Wellcome Trust. So this is a talk about Pseudomonas infections of cystic fibrosis patients. So as you probably know, cystic fibrosis, it's a genetic disease, and it causes individuals that have it to have a very thick mucus. And so one of the things that your mucus does in a healthy lung is to be beaten up by the cilia and to clear any infections. And because the mucus is very thick in cystic fibrosis patients, that doesn't happen properly. So they're very susceptible to infections. And there's one infection, Pseudomonas aeruginosa in particular, which is particularly nasty. So if the clinicians can get at it early enough, they stand a decent chance of clearing it, but quite often not. And rather rapidly, it will establish, and once it establishes, it's very difficult and impossible to get rid of. And it becomes a chronic infection, so these Pseudomonas infections can last several years, but they're associated with loss of lung function, and ultimately the patients either die or they need a lung transplant. So essentially we've been thinking about how we can view that process of chronic infection and try to understand what goes on within that infection in terms of the ecology of the bugs themselves. So the Pseudomonas itself, patients can acquire that in one of two ways. They can acquire that either from the environment. Pseudomonas is an opportunistic pathogen, lives a lot in the environment, but can get into barns or any susceptible individuals. But it also became apparent that there were transmissible strains of Pseudomonas. So these are strains that seem to be adapted to transmitting from one individual to the next. And one of the first strains of these found was called the Liverpool epidemic strain. And this is because Liverpool's particularly rich in dirty pathogens that we pass out to the rest of the world. It's because we had such good microbiologists there that found the strain there. And so this is found kind of around the world, but it's particularly good at transmitting between different individuals. So this is the study design that we have here. And so essentially 10 patients, teenagers, young adults within a cystic fibrosis clinic came in at the start of 2009 and then took a sample from those individuals. And taking a samples is quite non-invasive. Essentially, they just cough up into a hanky and take a sputum sample from that and gets passed to the microbiology lab. And then from that, there were 40 isolates taken from a plate once it was grown. And that's actually a little bit of a departure from what had happened previously. Typically in a clinical microbiology lab, they'll take one, perhaps two clones from a sputum sample to see whether it's antibiotic resistant or not, etc. And so what Craig wanted to do was to look at perhaps some of the diversity within these individuals, within these infections. And what they also did from the clinic was to take longitudinal samples from these 10 patients. So shown in green there is when they're nice and healthy and they come in at the start of the study, and other green samples there are when they're coming in for the regular checkups. And sometimes they'll come into hospital as well when they're ill, when they've got what's called an exacerbation, when they're ill and they need antibiotic treatment in hospital. And from these 40 clones that were taken, one can measure a number of different phenotypic traits for bacteria, so things like the morphology of the colony, the range of antibiotics that they're resistant to, whether they're producing exo products like piocyanin, whether they produce biofilms, and so forth. And so a number of those phenotypes were taken. And along with that, what we're doing particularly in my lab is to do genome sequencing of either single clones or of pools of these 40 clones from each of these longitudinal samples. And so we know from other work, particularly with the issue of the environmental samples coming into individuals, that you tend to get adaptations of those environmental Pseudomonas samples to the lung. But we're interested here in these transmissible strains, whether those were already adapted in the kind of diversity that you would get, or do they continue to evolve once they get into patients. these kinds of questions. So, I mean, it might be the case that, you know, they're already quite well adapted to living in the human lung, and actually you wouldn't see that much diversity or evolution thereafter. That's not what you find. So this is looking at the diversity within those samples. So this rather fancy figure here comes from eBurst, and so this is taking all of those phenotypic traits and asking how similar different clones are from each of those 40 clones within a sample from each of those 40-odd samples that were taken from 10 patients. And you find actually that about half of the diversity that you see in the total data set is associated with variation between clones within a sample. So clearly these cystic fibrosis patients, their infections, which they've had for several years, have actually got a lot of diversity within them. And so kind of the different colors here are showing resistances to different antibiotics, and so those seem to not cluster together within this eBurst diagram. Because we've got longitudinal samples, we can also look to see whether the set of haplotypes that an individual has within a lung actually changes through time. And actually that also seems to be very dynamic, and that's shown by this bottom graph here. So here, for example, are 6 patients, and it's comparing the similarity between the set of haplotypes, so the set of phenotypes that each clone has, comparing the similarity between pairs of samples within one patient. And you see a negative correlation there if you look at time on the bottom and haplotype sharing there, that samples that are close together in time tend to look more like each other, but that tends to decay off as you go through the days, weeks, and months. So it's kind of a continual turnover of different clonal types there within these infections. So after having done the phenotyping side of the work, we then looked to see what was going on the genomic level. So present, first of all, these data here, which come from a single sample. So I said that we took 40 clones from each of those sputum samples cuffed up into a hanky. And for one of those samples, we sequenced all of those 40 clones. And this is phylogeny that David Williams, postdoc, produced. And so one of the surprises from this is that you find this quite divergent, these pair of lineages here within a single patient. So there's a lot of SNPs separating these two lineages there. So that might suggest one or two different things. So it might be that very early on in that infection, there's an early evolutionary split once that initial infection gets into an individual, and then those two lineages are maintained through that chronic infection several years later to when we actually sample those individuals. Or it might mean that actually the process of infection isn't a single event, but that there might be superinfection events going on. So that was the next thing that we tested, because we've got these various samples from a number of patients all going into the one clinic. And so these are those two lineages that I showed before in blue and red there from that cystic fibrosis patient 3. And if you look at the clones that we picked out and sequenced from other patients, this tree is not consistent with a single source of infection and then diversification of that single clone in that patient. This patient appears to have picked up these divergent lineages probably at different times during that patient's history in order to be placed on different parts of the tree. And so these are all the one strain, but there's diversity within the circulating strain present within this Liverpool cystic fibrosis clinic. And this kind of makes sense. So I don't have data for these individuals, but quite often cystic fibrosis patients go to summer camps, they go to the same clinics together, quite often they end up in relationships with each other. So they're probably coming into contact with each other quite regularly. And, but it does highlight the need to try to control these, the passage of infection from one individual to the next. So previously I showed the phenotypic turnover that you find within an infection, and so this is using pooled data here. And essentially what this shows is that that turnover that you see in the phenotypes there, asking the question, do you also see a turnover of these divergent lineages? 1 minute, lady. One, okay. So you do find these, the turnover of these lineages within these infections. And here's some of the genes that are involved. You find parallel mutations in many of these antibiotic resistance genes, biofilm genes, and one consequence of that turnover is that actually you might have selection at one of these genes, but actually it pulls up all of the other genes within that lineage. So each of these circles here is one of these lineages and the mutations that are fixed within that. And since Ellie tells me that I'd run out of time, I'm going to just leave that conclusion slide up there. And if there's any time for questions, I'll take them. That's right. Probably we don't have enough data for that. But it also, I mean, it seems to be the case that within a patient, we don't necessarily see, a dominant type there. So at least half of the patients have got two coexisting lineages there. So, if they're a dominant one, you would expect a lot more of them just to have just that one dominant one there. So probably there's some process of competition there, and it might be that, they're adapting to different niches or whatever. But it's, but as opposed to Nicole's talk, it's much harder to address that within the lung of a cystic fibrosis patient than it is within a squash bug, unfortunately. It's because we've got such good microbiologists that can recognize them. So the question was, how can I be sure that the culturing approach was capturing all of the diversity within the sample? This is only focusing on Pseudomonas, and it's not looking at all of the other ones there. So they're tested for other strains of Pseudomonas using a kind of MLST type approach, and so these individuals just have this LES strain. Probably we've done qPCR of some of these other lineages there within the actual sputum rather than those 40 samples. We do find low levels of other lineages present within there, so presumably at a very low level, and then they seem to come up later on in the infection. But certainly within those 40 samples, doubtless we don't get all of the diversity that's there. It's just a bit of a snapshot. Oh, it's not working for me now. Sorry, yeah. So certainly, most of them have mutations in things like LASR, but there's still some diversity. There's still some that don't. There's still some of that variation there. So whether they're sort of in different parts of the lung or, you get cheats and et cetera in there, I don't know.
look, so I'm mobile now. Right, so our next speaker is Levi, who I've just closed my book, so I can't remember what your title is. There we go. Thanks very much.
All right. Okay, so I'm Levi. Stand over here. Today I'm going to tell you about a couple projects that I've been working on. This is going to be sort of a broad view of those projects. And I want to start out and just thank Ellie and Kayla and Zen for organizing this today. Thank you guys for coming out as well. First of all, I want to point out that this work was done with a massive effort by lots of people. I'm going to highlight some of them sort of as we go through. So even though I'm the one talking here, it doesn't mean that I even did the bulk of the work here. Okay, so we're talking about host and microbe interactions. And I think of these interactions as existing on this continuum here as somewhere between antagonism and mutualism. And in my lab, we focus on the evolutionary trajectories of populations. And I come at this sort of continuum from the standpoint of an interest of how host-microbe interactions influence the evolutionary trajectories of populations and how that might influence their position somewhere on this continuum. And so today, I'm going to come at two questions sort of with this in mind. And the first one is, how do host-microbe interactions transition from this level of antagonism over here to mutualism? And the second question I want to get at is how are mutualistic interactions maintained? So once you've got a mutualistic interaction, how do you continue to keep that mutualistic interaction there? And obviously these questions are far too big for me to answer right now. I think in the span of a career, I would be very lucky to be able to get an answer in these things. So I'm going to bite off just a tiny little piece today and probably not even fully capture that, but we will hopefully make some progress here. And so what I want to ask, specifically with regard to this idea of moving along this continuum, what is the role of coevolution in movement or maintaining your position in this continuum? And so the first question I want to focus on is this idea of moving from an antagonistic relationship towards a mutualistic relationship. And the work I'm going to tell you about today was really spearheaded by an amazing scientist named Amanda Gibson. She's a postdoc in my lab currently, and she certainly gets all the credit for what I'm going to show you here today. Okay, so in my lab, we use experimental evolution as a way to get at the evolutionary trajectories of populations. And so what we wanted to do is take an experimental evolution approach to understand how you might transition from an antagonistic host-microbe relationship towards something less antagonistic, so sort of moving on the continuum in the direction of mutualism. We want to ask the question, what is the role of reciprocal evolutionary change or coevolution versus what I'm going to call today single evolution? And what I mean by single evolution in this case is perhaps the host is only changing going forward in a particular group, or a parasite is only changing going forward in a particular treatment. And we're interested in sort of comparing when you have reciprocal change versus these independent changes in hosts versus parasites, what that yields in terms of the relationship going forward. And so what we're going to do here is essentially evolve hosts and parasites singly, and then we're going to pair them and say, how did the relationship change when you had the single evolution versus when we allow reciprocal evolution and we pair them up? And our hypothesis here is essentially that if coevolution is important, then we would expect for less antagonism to have evolved here in the coevolution group as opposed to single evolution. And so to do this, we're going to need a host, we're going to need a parasite, and in my lab that often means using C. elegans as our host, and this bright red bacteria here, Serratia marcescens, as our bacteria. So in most interactions between Serratia and C. elegans, it's bad news for the worm. What you can see here is a nice healthy worm hanging out, and this is an infected worm, this sort of bright red infection from Serratia, and it very rarely ends well for the worm. Serratia is incredibly virulent and can kill the worm very quickly. But what's interesting is in several strains in my lab, we actually see that the host can actually tolerate infection from the Serratia. And what I'm showing you here is this is an infected host up here on top, and then this is an uninfected host here on the bottom. And what you can see is sort of the marker for this tolerance is actually colonization of Serratia in sort of the upper gut region of the nematode. And that's what I'm highlighting here with this arrow. I'm going to tell you, we've photoshopped this to make it red, but it is a really nice dark band that you can see under the microscope. And in the case of my Serratia, the Serratia is actually quite red as well. So when you look at this under the microscope, it actually appears as though the worms have a red neck. And so given that we are in Georgia, we like to refer to this as the redneck phenotype. However, I will say that in terms of political correctness, we were a little bit hesitant to publish under that name. So if you find our paper, it is now termed the ruby-throated phenotype. And so I will refer to this as the ruby-throated phenotype going forward, but you all know what we call it in the lab. That's just be our secret. So we have this ruby-throated phenotype with the worms, and what we did here is develop a co-passage protocol where we take our hosts, we expose them to the parasite in the presence of their normal E. coli food. And we see some proportion of the hosts that actually develop this ruby-throated phenotype, and we actually select on those hosts and that particular bacteria going forward. And so we isolate the bacteria from these hosts and we transfer it to the next generation. And we take the offspring of these hosts and we transfer them to the next generation as well. So essentially both hosts and bacteria are rewarded under this scenario for generating greater fecundity in the worm. And greater fecundity in the worm thus leads to greater fecundity in the bacteria here. And so we did this for 5 replicate populations through 20 different generations of selection. And this was our co-passage experiment, our co-passage treatment here. We also, just as I was talking about before, we have these single evolution treatments. And so what I'm showing you here is we are selecting for the ruby-throated worms. And we take the offspring of ruby-throated worms, we passage them to the next generation, but every generation the bacteria stays static here. So we just continually reintroduce the ancestral bacteria. And so in this case, worm is allowed to evolve, bacteria is not. It's just the opposite situation over here, where we select the bacteria from the ruby-throated individuals, transfer it to the next generation, and introduce ancestral worm. In addition, we had controls where hosts did not see the bacteria at all, and we also had controls where the bacteria did not see the host. Okay, so what we're looking for here is a reduction of antagonism in this particular relationship. And so the way we're actually going to get at that is looking at the fecundity of ruby-throated individuals. So individuals that have this ruby-throated phenotype from these different pairings that I'm showing you here below. So right here is this is the ancestral fecundity of the host. So we take the ancestor bacteria, the ancestor host, we pair them together, we make the ruby-throated individuals. This is the fecundity that we end up with, okay? In addition, we paired our control lines. We took our singly evolved populations, we put them together to make ruby-throated individuals, and we look at their fecundity, and we also look at co-passage individuals here. Okay, what you can see is that in the co-passage treatment, we end up with significantly greater fecundity relative to ancestral control or singly passaged. And so from this, we can take away that co-passaging was required for reduced antagonism in this particular situation. But after this point, we wanted to ask the question, was this really coevolution? So we were co-passaging these things together, but do we actually have evidence that it was a coevolutionary relationship? And so to do that, we wanted to look at local adaptation, or to test for specificity with these these co-passaged host and parasite lines. And so what we did here is look at pairings where we took our different host replicates from the co-passage treatment and we paired them with different parasite populations that were also part of the co-passaged treatments. And so here along the diagonal, we have our sympatric pairings, where this worm population 1 was co-passaged with parasite population 1, but anything off that diagonal is an allopatric pairing, where there would have been no opportunity for specificity to evolve in those interactions. And so what we see here is, if we look at fecundity, just comparing allopatric to sympatric pairings, is that we see much greater fecundity in our sympatric pairings as opposed to our allopatric pairings. And so this is a strong indicator of reciprocal evolution in that co-passage treatment. And so we ask the question, does coevolution matter as we're transitioning from this antagonistic relationship towards a mutualistic relationship? And what we can say is that coevolution in this case is critical. Although, however, I want to point out that this is probably a gross over-exaggeration of what we actually got in the experiment. I would actually say that this is probably a little bit more accurate. So it looks a little bit wimpy, but still in the right direction. Okay, our next question I want to address is how are mutualistic interactions maintained, and what is the role of coevolution? And for the particular system that we're going to look at here today, I want to point out that we can't get at that question directly, and instead I'm going to tweak the question just a little bit, and we're going to ask, what are the consequences of single partner evolution? And so what we're going to look at is the nematode Cytonema carpocapsae and its mutualist bacteria Xenorhabdis. And so Xenorhabdis colonizes a vesicle here in the nematode, and together they parasitize insect hosts. And so this system is fairly nice for several reasons, but one is it's a well-established mutualistic interaction, but it can actually be broken apart in the lab, and that's exactly what we did. So here, slight limitation on the system. It is very hard to passage the host only, but you can very easily passage the bacteria through insect hosts via injection without involving the nematode in any way. But you can also co-passage it with the nematode. And a nice thing about this system is that after you've done this co-passage and this experimental evolution, is you can actually reconstitute the mutualism. You can take the bacteria that has evolved in the absence of the host, put it back into the host, and ask basically what happened to the mutualistic relationship. And so we designed an experiment here where we co-passaged both worm and bacteria, and a treatment where we simply passaged the bacteria only. And we wanted to ask about how do these respond to selection in their relationship versus apart from their relationship. And so in one treatment, we selected for greater virulence against their insect host, and in another one, we had a control where we didn't select for increased virulence. And so here where we're selecting for increased virulence, what I'm showing you on the y-axis is the mean time to death of the insect host. So low numbers here are going to mean higher virulence. And so what we did after experimental evolution is we took the bacteria, we took our bacteria out of the nematodes here, and we infected insect hosts to just look to see how they responded to selection for increased virulence. And what we see is that in our bacteria-only passage, we saw the strongest response, most rapid response to selection in that bacteria-only group, as opposed to when they were co-passaged. And so from this, we can see that single evolution accelerated the response to selection. However, what we found is that this came with some consequences. And so after we did this experimental evolution, we took the bacteria that were co-passaged and the bacteria that were passaged alone, and we put them back into worms, and we asked, how good are they at recolonizing the host after having been apart? And what we see here is that this group that was passaged as the bacteria only is actually a very poor mutualist partner, in that they have a very hard time reconstituting that mutualistic relationship. And so overall, what we found is single evolution in this case was very detrimental to the mutualism. Certainly when they were responding to selection, but even in the control where they weren't necessarily responding to selection, we see some degree of breakdown in that mutualistic interaction. Okay, so in this case, where we're trying to maintain a mutualism, does coevolution matter? Certainly in the case of the microbe, what we find is that single evolution can be very detrimental. There may be a little bit more leeway on the end of the host. The host may not suffer as many consequences from single evolution, but overall what we think is that the coevolution is very likely to maintain this long-term relationship. And so I want to get out of the way so Ellie doesn't tackle me here, but I want to thank everyone here in my lab and my funding sources. Thank you guys, I appreciate it.
Thanks very much, Levi. I think we've got time for one question, maybe. Kayla. Do you want to Otherwise,
Thank you, Levi. That was a great talk. Do you have any idea, for the first part of the story, what the worms and this ratio are doing to reduce their antagonistic relationship?
That's a great question. So I will say the straight up answer is we actually don't know what is occurring, sort of what the worm is doing or what the bacteria is doing. Right now, the data that we do have, though, shows that if you take I should say that the interaction is incredibly specific, so that if you take the bacteria that has evolved with one particular worm population and you move it and pair it with any other, I showed you the fecundity results there, but actually the virulence is incredibly high when you disrupt that specificity. So whatever is happening there, it's really, really specific. And so it's kind of a not great answer, but to the level that we know, that's what we know what's going on. Cool.
Okay, thanks very much, Levi. Clapping. So our next speaker is Anneliese, who is come over from Rennes in France, and she's going to be talking about microbiota and recognition systems in Drosophila.
Okay. First, is it possible? Yeah, thank you for the slide. So it's my pleasure to be here and back to Liverpool for a few days. And I'm going to talk about a recent experiment I've done in the lab, mostly in collaboration with Zen Lewis. And the title of my talk is Microbiota and Recognition System in Drosophila. So I'll begin by a quite short introduction on the impact of the microbiota in behaviors in insects. And then I will present you two studies I've made, one recent one on the impact of experimental evolution of diet specialization on mate choice, and the second one on the impact of the microbiota on kin recognition when choosing a mate. And I'll finish by a quick conclusion. So microbiota has impact on behavior in insects, and it's well known, for example, in fungi, such as the cordyceps, which zombify the ants, for example. And it's also well known in symbiotic bacteria, such as Wolbachia, which has been entitled the master manipulators of reproduction. And some other examples exist in Cardinium, which affect host choice in the parasitoid Encarsia, but also in Spiroplasma, which affect the cannibalism rate in ladybird larvae. And our question was, what about commensal bacteria? Commensal bacteria that are extracellular, that lives on the cuticle, the genitalia, or even some internal organs of the insect. And these bacteria, we know that they can affect the physiology, but also the nutrition and the digestion in insects. And quite recently, it has been shown that these bacteria could alter the mate preference in Drosophila. So in these studies, which are the beginning of our experiments, in some of them, the author cannot differentiate the effect of the diet and or the microbiota on the mating preference. And in some other studies, there's a confounding effect about the inbreeding, about inbred lines that were used in the tests. In all of these studies, anyway, they were using a character that is that the fly are grown in a specialist diet. So they always grow in the same diet over generation. And in Drosophila, so the bacteria, they are acquired from the diet, mostly, and also some of them are transmitted from the mother. But what we know is that, what we expect is that in specialist species who feed regularly on the same diets, the host microbiota association should be more stable and sustainable over time and over generation. And by contrast, in generalist species, we expect that the host microbiota association should be more transient over time, leading to a loose association between the host and the microbiota. So the induction of mate preference by the microbiota is more likely to occur in specialist species than in a generalist one. And here comes my first experiment on the impact of the experimental evolution of diet specialization on mate preferences in Drosophila. So we worked on 2 generalist species of Drosophila melanogaster and simulans, whose microbiota is supposed to change over time and with diet. And for these 2 species, we created 3 populations. So the first one is a generalist population, whose diet is alternated at each generation. And we also created 2 other populations, one specialist on corn and the other one specialist on banana-based diets. So for this specialist population, the diet remains the same at each generation. We let them evolve for 20 generations, at which point we, from our basic stock of melanogaster and simulants, we recreated these 3 populations that evolved just for 1 generation. And at these 2 evolutionary times, we took the female and we give them the choice between a male from their own population and a male from another population. We did that also for the specialist species. And we scored the number of homotypic mating, so the number of mating that occur between individuals from the same population. So this allowed to calculate the sexual isolation index, which is the number of homotypic mating minus the number of heterotypic one. So when the female mates with the male of another population, divided by the total number of mating. And to control for the impact of the microbiota on these mating preferences, we repeated all the experiments, all the tests, by using males that were axenic, so without microbiota. So here are the results for Drosophila simulans. On the y-axis, you've got the sexual isolation index. When this sexual isolation index is significantly different above the 0 line, it means that the population is sexually isolated. In green, you've got the corn specialist population. In yellow, the banana specialist one, and in red, the generalist population. And now, when the female had to choose between 2 males whose microbiota was intact, it's represented here in plain bars. And when the female had to choose between 2 males whose microbiota was altered during the development, it's represented here in the dashed bars. So what we can see is that we observe some significant mating preferences, which can be above the 0 line, so meaning that there is some sexual isolation here, but for some of them it can be below. And these mating preferences, they evolved from the first generation here, G1, to the 20th generation. And we observe that in all our 3 populations, so the corn, the banana one, and the generalist one. We observed that when the microbiota was present in the male, so this means that the microbiota associated with the diet can induce the mating preference evolution in these strains, in this population. But we also observed it when the microbiota was absent in male. So it means that diet alone also induced some evolution of the mating preference in this population. And more interestingly, we also found that for 2 populations, so the one at the 20th generation of evolution for the corn specialist female, what you can see here is that homotypic mating here are determined by the presence of the microbiota in the male. So when the microbiota is absent in the male, the female switch their mating preference for heterotypic mating. So in both of these cases, in the generalist population and in the corn one, it means that the microbiota alone can induce mating preference in this population. So now, concerning Drosophila melanogaster, the results are quite different, a bit less interesting, because we also observe some of mating preferences that are significant for the banana population and the generalist one. We only observed, however, an evolution of these preferences in the banana population from the first to the 20th generation, and whatever the presence of the microbiota in the male. And in Drosophila melanogaster, there were no effect at all of the presence of the microbiota in inducing mating preferences. So here it's a small table to summarize what we've got, and just to show that, in fact, in simulants, in all three populations, the corn, the banana, and the generalist one, we've got an effect of the diet that is different. We've got an evolution from the first to the 20th generation in all these three populations in affecting the sexual isolation. And we also observe an impact of the microbiota in these two populations. By contrast, in Drosophila melanogaster, there's some impact of the diet in the banana population and generalist one. And we only observe an evolution of the mating preference in the banana population. So from that, we can conclude that the sexual isolation we observed is quite multifactorial, and it varies with the species we studied, the diets, but also the diet regime of the fly that we maintain over generation, and for some of them with the presence of the microbiota. And it suggests that maybe that the microbiota could have some impact on the host pheromone that is used at mate choice. And here it's just to introduce the second studies we've done in collaboration with Stan Lewis and with Chloe, which is here too, where we studied the impact of the microbiota on another behavioral processes, which is kin recognition, so which is the fact, the ability to recognize related individual from unrelated one when choosing a mate. So in this study, we use a two-factorial design where we offer the male to mate with a sister or an unrelated female in a no-choice test. And these female, their microbiota was either intact or it was altered via an antibiotic. And we measured mating proportion, so the number of mating we observed, which is supposed to be female control in Drosophila melanogaster, and also copulation investment, so in terms of the number of sperm transferred by the male at the point of mating, which is male control, obviously. And we also went further to see if this has some fitness effect on the number of eggs laid by the female after. So from the female point of view, what we have is that in these no-choice tests, we measure the proportion of mating, and whatever the presence of the microbiota in the brother of the female, the proportion of mating is equivalent. However, and interestingly, when the male is unrelated, the proportion of mating with these unrelated male decreased largely when their microbiota was intact, so in the male. So it seems that the females that kind of avoid extreme outbreeding, maybe using the partner's microbiota to estimate or to assess the relatedness. And this is kind of supported by other studies which shows in choice tests that the female Drosophila melanogaster, they prefer to mate with a more related male than less related one. So now, from the male point of view, so we score the number of sperm transferred and also the number of eggs laid by the female after the mating. And what we've got is that the male, when they are mated with an unrelated female whose microbiota was altered via an antibiotic, they increase the number of sperm transferred. And this translates into significant higher number of eggs laid by the female. So the male transfers more sperm to unrelated female, and this has some fitness effect for the mating couple, but only when the microbiota is altered. So this suggests that it might exist a sexual conflict over kin recognition at the point of mating between male and female in this species. And it also suggests that the interaction, there's an interaction between the microbiota and the Q used at mate choice in this species, with female relatedness Q being somehow masked by the presence of the microbiota in the female. So we went into studying that by doing some gas chromatography coupled with mass spectrometry to study the smell and the odors of the females. And we focused on 14 cuticular hydrocarbons whose quantitative variation makes the female scent. And from these cuticular hydrocarbons, the analysis showed that the microbiota only impacts on these cuticular hydrocarbons. And these CHCs, their particularity that they are commonly expressed in both male and female in this species. So in fact, we've got less quantitative variation between of these CHCs between related female, but this interacts with the microbiota, which is masking it. So here comes my last slide about concluding on these experiments. Word within word, we show that the microbiota can have an impact on the pheromone of the females in our species. And this can have quite big consequences at the point of mate choice and also on the kin recognition expression in these species, having some effect indirectly on the fitness of the mating couple. And this could have also a larger effect on the population structure, but also on reproductive isolation and speciation phenomenon. And I'll thank you all for your attention. Thank you.
So we might have to move on to the next one. Yeah, that's all right. And then, so Annalisa will be around at coffee, so please stick around and attack her afterwards. So our next speaker is Isabel, yep, Gordo, who's talking about rapid bacterial evolution within the gut.
Hi. Okay, there's it. Thanks a lot for inviting me. It's a pleasure to be here. It's my first time in an ecology conference. So I'm an evolutionary geneticist, and I'll be telling you about the study of evolutionary change of a bacteria in the guts of mice. And I'm located in Portugal. So this is just to remind you that we can't eat properly without the microbes that we have in our gut, and the ecosystem that we have inside us has many species and encompasses many interactions, which makes it very hard to understand. Importantly, there are strong microbe interactions, which we can start to understand when we perturb the system and follow how each of the groups of bacteria changes through time, and also important interactions between microbes and cells of the adaptive immune system. And in the two studies I'm going to be telling you about, these interactions will be important. Okay, so we are interested in understanding the pace and the mode of evolution of a commensal bacteria within a vertebrate host. And we picked a mouse and a very nice bacteria, E. coli, that can grow very well everywhere. And it's also one of the first colonizers of us when we were born. And we want to understand what evolutionary mechanisms dominate the evolution of this bacteria. Is it natural selection or genetic drift? And what kind of selective pressures are important in this environment? So we use the evolution of the organism to tell us something about the environment, the experiences. And in this sense, we want to understand how does the adaptive immune system influence its evolution. So our protocol, our system is to take an E. coli that is isogenic, so it's a clone, but we have two forms of this clone. In the genome, we put a fluorescent marker, so we have a clonal population with a neutral fluorescent marker, such that half of the population fluoresces blue, half fluoresces yellow. So we are interested in understanding the acquisition of the novel mutations on this lineage through time. And the way we do this is that we have to give the animal an antibiotic so that E. coli can go in there, so that it kills the resident E. coli, and the one we want to study is resistant and can colonize. And then every day we sample the feces of the animals and ask, is this neutral marker going to change in frequency through time? And we want to know how fast does it take for that to happen, how predictable is adaptation, and what adaptive mutations occur. Okay, so we can have a very simple scenario where mutation is a limiting process. It takes some time for a mutation to emerge. If it emerges on a background, it will sweep to fixation. But we can have a more complex process that is pervasive in every microbial experiment that we do in their laboratory conditions, which we have multiple beneficial mutations occurring and competing, giving rise to the process known as clonal interference. So what we actually see when we follow 15 hosts, the bacteria colonizing 15 hosts here in different colors, is that within less than a week, we can see that sometimes we have a rapid change in frequency of the yellow ones, or a complex marker dynamics compatible with clonal interference. Now, a process like this could be the result of genetic drift if we have very few bacteria actually inside the gut. But if it's really adaptation, then the bacteria that we can sample from here, if we take them and compete them against the ancestral, they should consistently outcompete the ancestral. And that's what is in this graph here. We see that for every evolved group of bacteria or single clones that we compete with the ancestral, they always outcompete by about implying quite a large selection coefficient. So with this data, there's clear that there is rapid adaptation driven by clonal interference in the guts of these mice. Okay, so then to know which mutations happened, we took a single clone that increased in frequency and sequenced its genome. And what I wanted to notice here is that there's a particular region, a particular operon that got targeted by mutation in all the independently evolved populations. And so here is a blow-up of the operon. And whenever there is a triangle that's a transposable element insertion, so basically what happens is that the first adaptive event is actually a loss of function. So it's killing this operon. And to have an idea on the pace of evolution, a clone sampled after about a month carries 2 to 3 mutations. So there is basically massive parallelism at the level of gene and operon in these experiments. And the adaptive changes have to do with the ability of the bacteria to change its metabolism and its respiratory capacity. There is also quite an important role of transposable elements driving this adaptation. So when we look inside a population and we take, say, after a week, a sample of clones and sequence that operon, what we find is that there are actually many different clones that carry mutations in that operon, and these clones are selectively equivalent. So what we have here is a very high rate of production of these loss of function adaptive mutations, each allele having the same selection coefficient. So we have a lot of genetic variation coding for the same phenotype, and the phenotype is increasing in frequency as a classical way of a selective sweep. So because what the bacteria that is evolving is doing is to lose the ability to consume a given sugar, in this case galactitol, we basically ask the question, if we change the diet such that we give the mice galactitol in its water, can we protect the function or dysfunction in the bacteria? So what you have in here is a competition between the ancestral bacteria that can consume the galactitol and one of the evolved clones that can't. And what you see, if you don't supplement the diet, you get a decrease in frequency of the ancestral, right? And it doesn't really have to get completely lost, so it can be maintained at low frequency. But if you supplement the diet with a little bit of galactitol, you can actually control the frequency of this strain in the animal gut. So it's quite strong selection, and our interpretation is that this is a limiting resource that is the bacteria consumed, then it's gone, and then it's no longer advantageous to have the function. So then we ask the question, what is the importance of the adaptive immune system for the evolution of these commensal bacteria? So for that, we did exactly the same experiment, but now we have animals that have a mutation such that they lack T and B cells. So this mutation, unfortunately, can also occur in humans, and these people can only live inside a bubble. So they would be in trouble if they would be in the natural environment. But the mice in the animal house live very happily because the conditions are very clean. So we actually can study the evolution of these bacteria in mice that lack an adaptive immune system. So what happens now is that when we look over 1 week in the immune competent mice, we could see the signature of adaptation, but in the immune compromised, we hardly see any change. So this data indicates that the pace of evolution is slower in the mice without an adaptive immune system. If we wait longer, then we see that adaptation also occurs in these mice. It's just we need to wait more time. So when we ask about this first step of adaptation that had occurred, that we had seen in the wild-type mice, we see that in the immune-compromised animals, there is far more variation in the dynamics of spread of this beneficial mutation than there is in the wild-type animals, suggesting that the presence of an adaptive immune system is creating coherence between the animals for this phenotype. So why is the rate of adaptation slower in the immune-compromised mice? Well, it could be because the population size is smaller, because the bacteria have a lower growth rate, have a different mutation rate, or actually selection strength is different. So in terms of population size, we don't see any difference between the bacterial loads in the mice that are immune competent versus immune compromised. In terms of growth rate of the bacteria, we actually see that in the immune compromised mice, the doubling time is smaller. So this is actually against the slower pace of evolution that we observe in these mice. In terms of mutation rate, so we tested for the occurrence of spontaneous mutations towards resistance to certain antibiotics in the bacteria, in the two animals, and we don't see any significant differences in the rate, in the frequency of spontaneous mutation. So we don't think the mutation rate is the cause of the difference in speed of adaptation. As far as selection is concerned, the result is very clear. So we act when we take new mice and compete the evolved clone with the first beneficial mutation against the ancestral. We always get basically a selective effect of about 6%. But when we do the same experiment in different reg animals, we actually have an average selective effect of the mutation that is half of that in wild-type animals, and far more variation in selection effects between the immunocompromised animals. So it seems like the different stresses of selection are the reason for the slower pace of adaptation. So in order to, because in the animals that do not have an adaptive immune system, they will also change their microbiota composition. We basically tested for the selective effect of this mutation after we co-house the animals so that we can normalize the microbiotas of immune-competent and immune-compromised mice. So they were co-housed for 2 months, and then we performed the experiment. And now what happens is that we get more variance in the immune-competent mice, and they become very similar. And the other thing that we did was to test for the effect of this mutation in the complete absence of microbiota, so that's germ-free animals, and here the effect is the same. So basically, it's the influence, it's the rest of the microbes that are the cause of the differences that we see in between immune-competent and immune-compromised animals. So in fact, when we look through the 16S to the microbiota of these animals, we did find that there is, in the immune-compromised, there is significant differences in the relative abundances of specific groups of bacteria, and we tend to see for some groups far more variance in their abundances. And so that's coherent with the phenotypic effects of mutations that we were measuring. So when we basically take large samples of the evolved E. coli from these animals and ask about which targets in the genome were changed in one host or the other, we find that there is a lot of targets that are common in between the two hosts, but then there are targets, for example, this one here, that only occurred on the immune-competent animals and never in the immune-compromised. And in this system, it seems that mutation is non-limiting, so we attribute this to differences in selection. So there's the specificity of the targets of the adaptation with the host genetics. So to conclude, I show you that the evolution of a commensal microbe can be very rapid in the mammalian gut. I wonder how rapid it is inside of us. I showed that the first step of adaptation that we observed was caused by distinct alleles causing the same phenotype, and that the pace of adaptation was slower in immune-compromised mice, and that there are some targets of adaptive mutations that depend on the host genetic background. So it's the interaction of the adaptive immune system with the other microbiota is influencing the tempo and mode of these commensal bacteria in the gut. Thank you.
Thanks very much as well. That's really awesome work. So again, I'm afraid we're going to have to move on to our next speaker straight away, who's Katie Field. And she's going to be telling us about the conquest of land through symbiosis. Awesome.
Let's hope this will work. Oh, goodness. Oh, sneak previews. Okay, cool. So thank you very much. So firstly, thanks Ellie and Zen and Kayla for inviting me here today. Really cool to be contributing to this session, which I think has been really fantastic so far. So thanks very much for organising that. So I'm going to be talking to you today about how plants first came onto the land and how they might have done that in symbiosis with symbiotic fungi. So in order to do that, I'm going to take you through the land plant phylogeny, which I'm sure we're all well familiar with. And this is the generally accepted version of the land plant phylogeny at the moment. And I know there's some controversy about the bryophytes at the end here, but we're not going to go into that today. We're just going to go with bryophytes coming first. So the very first plants came onto the land around 450 million years ago in the Ordovician. And these plants were most likely liverwort-like, without roots or vascular systems. And they were followed up quite quickly by the other bryophyte clades, and then followed by rhiniophytes, which are now extinct. But we know that they are existent because of the fossils taken from the Rhynie Church up in Scotland, and followed by vascular species that are still extant today, including club mosses, ferns and conifers, and obviously the angiosperms. Now, one of the common features across the entire land plant phylogeny, which I've illustrated on this figure, is that each of the different clades, apart from mosses, associate with mutualistic fungi known as arbuscular mycorrhizal fungi, which belong to the Glomeromycota. Now, these soil-dwelling fungi are obligately biotrophic, and they exchange nutrients from the soil, which they're able to forage for through their extra-radical hyphae, which they're able to exchange with land plants for the photosynthase they produce through photosynthesis. Now, as illustrated in this, we kind of have assumed what is the prevailing dogma has been that the earliest land plants were able to colonize the land in the first instance 475 million years ago, thanks to the partnerships between them and their mutualistic soil fungi. We have to consider those earliest plants didn't have roots, and they were very limited, therefore, in the way that they could scavenge nutrients from the soil, which was practically non-existent at that point. There was very little organic matter in that soil and little in the way of plant-available nutrients. And so it was this partnership between plants, and it was the fungi of the Glomeromycota were thought to have been what allowed plants to colonize the environment in the first place. However, in 2011, Martin Birartondo and colleagues published this really important and exciting paper showing that actually the most early diverging lineage of liverworts, itself being the most early diverging lineage of land plants, were actually not associating with fungi of the Glomeromycota at all. So they weren't associating with arbuscular mycorrhizal fungi. Instead, he found that they were associating with partially saprotrophic fungi of the Mucoromycotina. Now, Mucoromycotina is a lineage of fungi that predates the Glomeromycota. So it kind of put this lineage of fungi in the picture as actually being the most, actually being the family of fungus that helps plants onto the land in the first place, which has kind of turned that entire hypothesis on its head and made us consider that perhaps the initial terrestrialization of land plants was facilitated not by arbuscular mycorrhiza, but by Mucoromycotina fungi instead. Okay, so that kind of changes our picture of the land plant phylogeny somewhat and the fungal associates that we map onto it. And I've put little red dots on the clades of extant land plants that we now know associate with Mucoromycotina. And you can see it's not just limited to these early diverging clades, but it's spread right throughout the land plant phylogeny. And there was a paper published in New Phyt only a couple of months ago, actually suggesting that angiosperms also associate with these Mucoromycotina fungi. So this is a really widespread symbiosis that's just previously unheard of before 2011. So all of this major land plant divergence, to put in the context of what's going on Earth at that time, was occurring against a background of declining CO2. So atmospheric CO2 at the time of the first land plants is likely to have been up in the thousands parts per million, and it declined sharply as land plants diversified and became more complex in structure and physiology. So this kind of leads us to the key questions for some of the stuff that I've been doing recently. And that is, are these associations between these early divergent clades of liverworts and this early diverging clade of fungi, are they functionally equivalent to the associations we see between plants and arbuscular mycorrhizas today? So are they mutualistic? And does that support the hypothesis that it was that type of fungus that helped plants onto the land in the first instance, rather than the arbuscular mycorrhizal type of fungi? And secondly, just to have a look at what the influence of atmospheric CO2 is on that relationship. Does that change the mutualism between plant and fungus at all in the same way that we've seen it affects the Glomeromycotin symbioses? So in order to do that, we have to go and collect liverworts from the wild, which have got their fungal partners already living inside of them. And these are our two species that associate with Mucormycotina fungi. We have Troibia lacanosa and Haplomytrium gibsei, both of which are very lovely, very pretty little liverworts. And we have to go to the South Island of New Zealand to collect them, which is, it's hard for me to go and do that. And the other two liverworts that we use to compare them against are ones that associate with Glomeromycotian fungi. So these are Pricea quadrata, which is collected from the less glamorous location of Anglesey, and Marcantia palliaceae, which we have in culture. So while we were collecting those plants and carrying out the experiments, there was another really exciting paper in the area came up, and it kind of changed things again. This picture is always changing. And it was this one published in the New Phytologist by Christine Strulideri and Natal. And they are palynologists, no, not palynologists, sorry, they're paleobotanists, they'll kill me for saying that. They're paleobotanists, and they were re-examining some of the Rhynie chert fossils. And in these fossils, they found what they were expecting, some of the fungal structures within the plant roots and rhizoids. So we're finding fungal structures that were similar to Glomeromycotian type structures, but they were also finding structures that are really similar to Mucormycotina fungal features that we've characterized in extant lineages. And most excitingly, they actually found some of these things, some of these structures appearing in the same plants at the same time. So this is adding weight to the hypothesis that Mucormycotina were key players in the terrestrialization of land plants, because not only are they present in the fossils at the same time, but they're appearing around the same time as Glomeromycotian fungi. So not only does it reinforce that hypothesis, it makes it even more complicated. They're both around at the same time, potentially doing the same job. So this changes our phylogeny somewhat a little bit more. So we're able to put a little red dot on the rhiniophytes, suggesting that they also associated with both types of fungi. But crucially, for what I'm interested in, what are they doing in there? What are these plants that are associating with both types of fungi doing? And luckily for me, some of the liverworts we collected from New Zealand in our last trip, Neohudsonia mirabilis and Allosonia cocknei, they both also associate with both types of fungi at the same time. So that gave us a suite of plants with their native fungal partners, ranging from single colonizations of Mucormycotina and Glomeromycota, but also these plants that had both of those symbionts present simultaneously. So we wanted to test whether these dual fungal symbioses were also mutualistic and whether they were also CO2 responsive or not. So the experimental setup that we did, I'll just talk through that because it's a little bit complicated, but I think it's important. So we grew the plants, we collected them from the wild and grew them on sand in these pot systems. And into the systems we introduced these mesh-covered windowed cores. And the mesh covering the windows in those cores is coarse enough to let fungal hyphae grow in, but too fine for the rhizoids of the liverworts to get through. So we effectively create like a fungus-only zone within the center of that core that only the fungus can access. And this allows us to measure the amount of carbon that is fixed by the plant and transferred to fungus growing within the core by releasing a 14 CO2 pulse into the system and allowing the plant to fix it through photosynthesis, and then we chase the fate of those photosynthetes through time. To look at the exchange for nutrients, we inject radiolabeled phosphorus and 15N into the center of one of these cores in each pot, and then we either leave that core static to let the fungal hyphae have access to the isotope, and then we're able to measure the uptake of the isotope and track it in the plant tissues itself, or we rotate the core, which severs the hyphae and provides us with a control treatment to test against. So we've either got fungal access to the isotope or not in this case. So if we look at some of the data we collect from those experiments, so I'll look first at the carbon transfer from the plant to the fungus. And apologies for these figures being so busy, but I can't think of a more simple way of presenting it. We've got single Mucormycotina-associated liverworts here. In the center panel, we have Glomeromycotian-associated liverworts. And on this panel, we have the dual association liverworts. I should have said earlier, actually, we carry out those experiments at ambient CO2, and we also carry it out at a replicated Paleozoic CO2 concentration of about 1,500 parts per million CO2 to test the effect of the CO2 on the symbiosis. Anyway, so the carbon transferred from the plant to fungus, and this is the amount of carbon that we measured in those cores at the end of the labeling period. The interesting pattern that all plants fixed more or transferred more carbon to their fungal partners under the elevated CO2 concentration, However, most interesting perhaps is that the plants associated with both fungi at the same time transferred significantly more carbon to their fungal partners under both the elevated CO2 and the ambient CO2 concentration. So it appears that it's kind of more expensive, if you like, to support two fungal partners rather than one on its own. If we look at the nutrient uptake from the fungus to the plant, we get quite an interesting pattern emerging. So all plants, well, the stop-press moment really was this with the single Mucormycotina colonization. So this was the first evidence that we ever had that actually it was a mutualistic relationship between plants and Mucormycotina fungi in this case, where we have nutrient for carbon exchange happening quantitatively measured. But interesting patterns emerging that under elevated CO2, these Mucormycotina associations are actually passing less nutrient onto their plant partner than under the ambient CO2 concentrations. And this is exactly the opposite pattern to what we see in plants associating with arbuscular mycorrhizal fungi, where they're actually getting more nutrients under the elevated CO2 concentration compared to the ambient. The dual associations, again, it's a slightly different story. They seem to follow the pattern more of a Mucormycotina-like association, where you get more nutrient uptake under the lower CO2 concentration. However, you can see it's quite a lot more. It's a substantially greater amount than under a single fungal colonization. So if we look at the nitrogen uptake, we can see a similar pattern where we've got more nitrogen being transported to the plant from the fungal partner under the ambient CO2 compared to the elevated CO2. But this was more, so it was more exaggerated, and it was also quite strongly significant in the dual colonization rather than the single colonization. So these plants that are hosting more than one fungal partner at a time appear to be gaining more from their partners, but it's also costing them more in terms of the carbon they're having to invest. So from this data, we're able to calculate a carbon for nutrient exchange metric, which we call efficiency, and it quite literally just means the amount of phosphorus gained per unit of carbon invested. And you can see the main point I want to say here is it's an opposite pattern again. Glomeromycotian associations, they operate with a far greater efficiency under the high CO2 versus the low CO2, and the opposite seems to be happening when a Mucormycotina partner is involved. Same thing with the nitrogen. So why have these dual associations persisted for so long? It appears they're much more efficient at the lower CO2 concentration than either of the single colonizations, which gives them a physiological niche through evolutionary history. It's also very possible that the plants in our systems were nutrient limited. They were in pots, which are mostly filled with sand after all. So maybe then they weren't able to utilize those excess photosynthase they were generating at the high CO2 concentration, remembering that they're liverworts and they don't have vast amounts of storage space for starch within their cells or within their tissues. So it may well be that under these high CO2 situations where the liverworts don't have access to nutrients, so they're able to utilize that carbohydrate for growth, that they actually offload it to their fungal partner. And this kind of then stimulates increased fungal growth. And in turn, that means the fungus has a greater nutrient demand itself, and so it has less nutrients transferred to the plant partner. And it may well be that actually this is a safe tactic for the tiny liverworts in a system which is dominated by saprotrophic organisms that would quite happily feast on these carbohydrate exudations into the soil and potentially outcompete them. So maybe better to pass your excess onto a friendly partner than some sort of competitor. And it may be that this explains our carbon for nutrient exchange data that we saw in our systems. So I think the main message I want to get across today is that actually the origins of these plant-fungal symbioses are far from resolved. These Mucormycotina associations are far more prevalent than we previously thought, and I suspect there's other fungi out there as well that we have not even considered yet. And it may be that this is an ancestral condition, or but from what we've done, the physiological evidence on its own is not enough. We need to be using fossil, cytological, and molecular evidence. But I think we can safely say that fungal symbioses are far more variable and transient than have been assumed thus far. And it may well be that these dual fungal associations are actually really important ecologically, and they're far more widespread. And this has implications for our understanding of plant-fungal symbiosis and evolution. And I'll breathe now. Thank you very much. It's collaborative work. I should probably mention like Martin Bedotondo, who does a lot of the molecular work for us, Sylvia Pressel, who does all our cytology, and Jeff and Will, who have been immensely helpful on the project. So thank you very much.
Thanks so much,
Katie.
I'm so cheeky. Look, my job advert up. If anyone wants to work in my lab on this project, then we've got a postdoc available for 3 years, so come see me.
Amazing. Well, we've been super data efficient, I think. I'm going to say that's a good thing. But so we'll move on to Olivia, and then we're going to have an awesome coffee question session after this. So Olivia's going to be talking about pregnancy in men, which is brilliant, and the influence of microbes in that. Thanks.
Thank you very much for the invitation. So males and females are different. And actually, in a traditional family view, females were responsible for the upbringing of the kids, while males went to work and, in best case, brought back home the money. This view has changed due to gender equality. But still nowadays, we actually face the problem that women are often much more attached to the offspring than men are. And the reason for this probably lies in the biology. So it's not that the females do have the cost of the large egg production, but in addition, they also have the cost of the parental investment during the pregnancy. This is a very high cost and also takes them a long time per reproductive unit. But on the other hand, the males only have to produce a small sperm and can then continue with additional mating if they want. So basically, parental investment and the most extreme form of parental investment, pregnancy, has evolved several times independently in vertebrates. However, we don't know exactly how it evolved and why it has evolved so often. And to study this, we face one issue, because actually all these events are female pregnancy events. And here, the costs of the egg production and the parental investment are all intermingled in the female. So we cannot exactly separate what is involved, what is important for egg production, so what is the sex difference, and what is actually the extent of the parental investment. And for this, there is one solution, which is the unique case of male pregnancy evolution in the animal kingdom, in sygnatids, so in pipefishes and seahorses. And they actually offer the advantage that we can disentangle sex from parental investment, which means that here, the females are still the ones that are producing the large, costly eggs that they then transfer to the brood pouch of the male, where the male is giving all the investment during the male pregnancy. So we can disentangle sex from parental investment. And this aids us to understand some life history adaptations that were said to be due to sex roles. And one trait here is sexual immune dimorphism. So what we see usually in the animal kingdom is that usually females are having a more efficient, a stronger immune response than males. And this was said to be due to this difference in egg production costs and in small sperm provisioning. However, when we look at this now in pipefish, then what we see is that this pattern changes. So here the males are the ones with the more efficient immune response than the females, which asks for parental investment to potentially play a role. And that's what we then investigated. So the nice thing about pipefishes and seahorses is that they evolved on a paternal investment gradient. So the more basal species, like the Neurophina here the females are producing the eggs and they transfer them to the belly of the male where they just are glued on. In comparison to the Signatus and Hippocampina, they do have a placenta-like structure, so a real connection from the father to the offspring. The paternal investment in the Neurophina is much lower than here on this side. So we can actually study the importance of paternal investment, of parental investment in sexual immune dimorphism. And that's what it did here. So we compared immune defense of females and males of these 3 different Baltic species. And what we find fits exactly our hypothesis. So here in the Neurophis, in blue, we did not find any sexual immune dimorphism. And this actually makes sense because potentially the cost of egg production on the female side is about the same as the cost of paternal investment on the male side during the pregnancy. Whilst here, in those 2 species, with actually a placenta-like structure, the cost on the male side is much higher. And this is reflected in the sexual immune dimorphism. So in those two species, the males are the ones with the more efficient immune response. This is part of the PhD from Isabel Keller. So this actually brings us back to the hypothesis that it's not important what sex you are, but it's just important how much you invest in your offspring. And actually that efficiency of immune defense is correlated with the extent of parental investment given. Another life history trait that is intermingled in this problem with egg production and parental investment is the transfer of immunity from parents to offspring. So it was said to be mainly the mother that boosts the offspring immune response in humans, and we all know this during pregnancy and breastfeeding. However, also here, potentially via the egg, there are already substances given over that might induce the offspring immune system. And when we study this in the pipefish, where we either immune challenge the female or the male, or both of them, then we see an increased immune response in the offspring, independent of which sex or whether both of them were challenged. So it really seems that both the egg production, but also the parental investment over the male pregnancy has a role in grasping the immune response of the offspring. And more importantly, it really seems that pipefish are somewhat sharing the task in how to boost the offspring immune response. So while an immune challenge of the female resulted in an upregulation of the adaptive immune system of the offspring, so via the eggs here, an immune challenge of the male resulted rather in an upregulation of the innate immune system of the offspring. So male pregnancy can be advantageous. And this is part of the PhD thesis of Anne Bemelmans. Similar to transfer of parental investment, we finally come to the microbe. Sorry, took a bit long. And basically, this is also a very important life history trait, the transfer of microbes from mothers to offspring. And we know that it actually makes a difference whether you have given vaginal birth or cesarean for the initial microbial colonization of your newborn. And also here, we wanted to know whether we can actually disentangle the microbial transfer from via the female or via the male. So what we did just as a first study is that we genotyped the microbial community on the eggs, still in the females, so this is then called gonads, the GO, or in the pouch of the non-pregnant male, or we genotyped the microbes over pregnancy during early pregnancy, mid-pregnancy, or late pregnancy. In addition, we wanted to know whether this transfer of immunity, so whether the maternal or paternal immune status somehow interacts with the microbial community. And doing this, we actually found that we have a distinct microbial community on the gonads, and then a very similar microbial community in the non-pregnant, so just in the brood pouch of the male, but also during early pregnancy and mid-pregnancy. However, during late pregnancy, where potentially the colonization of the microbial community occurs in the offspring, we find a distinct community and much more variation in the community of so during late pregnancy. So there's something happens. And actually, we can find, when we looked at these index species, we can find specific contribution from the mothers or from the fathers. So we have several species of bacteria or strains of bacteria that are actually specific to only occur in the gonads or only in the non-pregnant brood pouch. And these are exactly those species that are then expanding during the late pregnancy, so they might play a role in the initial colonization of the offspring. But however, this was just the main effect of the tissue, so whether gonads or whether it's brood pouch. When we now include the interaction with the immune challenge of the parents, then what we find is that there is not a major influence on the microbiota neither in the gonads nor during the non-pregnant, mid-pregnant, or early pregnant stage. However, then in the late pregnancy, where potentially this colonization takes place, we see that the immune status, here late pregnancy injected versus late pregnancy control, really matches, and that there is a difference in the microbial community that we could have genotyped. So this means that there might be a major influence of the parental immune system on the microbial colonization of the offspring. To end, I also want to point out some adaptations that might be important in the evolution of pregnancy and compare them between female pregnancy and male pregnancy. So when we look at the immune system again, one of the most important factors that has to be fulfilled is actually the immunological tolerance of the offspring, as the issue is that the maternal immune system sees the embryo as non-self, and the immune system would be activated and reject the embryonal tissue. In humans and in mammals, there are several traits that have evolved to prevent this rejection. And one of them is, for example, relying in the placenta. So we have here this layer of trophoblasts, and they do not express the very variable major histocompatibility complex 2, and they only express non-diverse MHC1. And this somehow prevents the recognition of the maternal immune system, that the maternal immune system recognizes the embryonal immune system. Interestingly, now, when we sequenced the transcriptome and the genome of Signatus typhus, of our model pipefish species, what we found is that both in the transcriptome and in the genome, there's absence of the major histocompatibility complex 2 pathway. So they have lost this pathway. And we now speculate, and we'll follow this up, whether it's actually this loss of MHC2 that facilitated the evolution of this unique male pregnancy. Many genes were co-opted in their function for the evolution of pregnancy, so neo-functionalizations have occurred. And just as a very short overview in how it happened, how this occurred in the mammalian pregnancy, so in the human pregnancy. So these are just the pathways, and of course many genes are behind those pathways, which I won't mention. So we have here trophoblast interferon, and the upregulation of this results in a differential regulation from the inflammatory Th1 to the anti-inflammatory Th2 response. And this also supports this immunosuppression so that the embryo is not rejected. In addition, on the trophoblast, as I said before, we have no MHC2 expression and only an expression of non-variant MHC1. However, this trophoblast interferon then also regulates the upbuilding of the endometrium and results in a downregulation of prostaglandin, which inhibits the degradation of the corpus luteum, which then over the progesterone pathway, the mTOR pathway, results in the implantation of the egg. There are many other genes involved for the embryo growth, for the placenta building, and for the transport from the mother to the offspring. When we do now a differential gene expression analysis with 48 transcriptomes comparing non-pregnant and pregnant pipefish males, then what we see, surprisingly, is that very similar genes are differentially expressed in male pregnancy. So actually, we do find genes that are known to play a role in all those different pathways that have a function in female pregnancy. And over 100 genes were potentially simultaneously co-opted both in female and in male pregnancy. So to summarize this, I hope it could show you that parental investment actually matters for key life history adaptations. So it's not just being a male or female, but it's really whether you are giving the parental investment or not. In addition, I think it's really surprising that male pregnancy has only evolved once, as actually can give you a double benefit. So while in a normal pregnancy event, you mainly profit from the maternal effects, here in male pregnancy, you may profit from the phenotypic, from the environmental experience of the mother and also of the father. So our initial data about the microbial communities show that there could be potentially an interaction of the immune status of the parents with the microbial colonization during late pregnancy. And we will follow this up now with manipulation experiments to really see if this matches. As I said, we are hypothesizing that the loss of MHC2 facilitated the evolution of male pregnancy. So for this, we have now sequenced 14 genomes of Signatiformes of either families that of either species that do not show male pregnancy, or of the Signatidae that do have this male pregnancy. And with comparative genomics, we can now compare the immunological reorganization that came with the evolution of male pregnancy. And as I could show you in a very short time, is that co-option of homologous genes has occurred in female and male pregnancy. And I think that this is really underlying that pregnancy is one of the most impressive examples of convergent evolution. And with this, I want to thank the people that are involved in this work, Thorsten, Anne, Till, Isabel, and David, and my group, and the funding, and you for your attention.
Thanks very much. Since this is the last one, we can probably have one cheeky question if anyone has one. Or we can all have a big coffee.
I think everyone is
I
tired.
think we should do that. So thank you very much to all our speakers who've come from all over Europe and further afield. So can we just give them a little round of applause? And for people interested in microbes, I want to plug a meeting that we've got tomorrow following some of this really cool work up at the University of Liverpool. It's in your brochure. It's called World Within Worlds, and it's tomorrow morning, starting from 9. So the web address is in there as well. Thanks very much.